Dr. Wintermantel, you are up again with Cucurbit Chlorotic yellows virus.
So this is the second of the what I would call the true yellowing viruses that we'll talk about today. Cucurbit chlorotic yellows virus is become an important part of the disease complex in the United States and many parts of the world. Cucurbit chlorotic yellows virus, or CCYV as we call it, has been emerging very, very rapidly in the US since it was first identified. It causes a severe leaf yellowing of crops and is particularly, again, like CYSTV, very important for production in areas where melons are produced, largely because of the impact on fruit quality. CCYV, like CYSTV, begins with a chlorotic spotting pattern on young leaves near the crown. These become coalesce into larger areas, what I would call a chlorotic mottle, like you see in the picture on the upper right. This eventually evolves into a symptom that looks, again, very much like CYSTV symptoms or a magnesium deficiency, with an interveinal chlorosis with the veins remaining green. This is very characteristic. The symptoms begin near the crown of the plant, as Scott just mentioned, and for the plant that he showed with the co-infection, and also spreads down the vines, much like CYSTV. So you see this gradual progression over time of this yellowing spreading from the crown toward the vine ends on a melon, a watermelon, or any other kind of cucurbit plant. Plants that become infected young can become severely stunted, particularly when they are infected at the cotyledon or first leaf stage, and fruit may be smaller or with reduced sugars. Plants generally have less vigor. CCYV was first identified in Japan in the early 2000s. The virus spread throughout parts of East Asia as well as the Middle East. It has been found in North Africa and Central America as well. The virus has, like CYSTV, not been found in South America or Australia. Within the United States, CCYV was first identified in 2019 in Southern California and Arizona. The interesting thing is the virus was actually present much longer. Because the symptoms are identical to those of cucurbit yellow stunting disorder virus, it was missed for several years until some breeding material began to show symptoms that it should not be showing. It made us suspicious there was an issue with the breeding material, but it led to the identification of CCYV, which had been present based on freezer samples, testing material out of our freezer. We found out the virus had actually been present since 2014 in Southern California and ultimately that Southern California, Arizona area. The virus rapidly spread to other parts of the country, was identified in 2020 in Texas, Alabama, Georgia, and Florida, as well as in the Central Valley of California. So once it spread, and it went everywhere all in one short period of time. CCYV is transmitted by two variants of the sweet potato whitefly, MEM1 and MED, also known as B and Q. Transmission differs between these two whitefly vectors. Acquisition, where the whitefly is feeding on the infected source plant, can occur within a couple of hours, but most efficiently for the virus to be transmitted effectively, the whiteflies need to feed a much longer period of time, generally 48 hours feeding. So you do have a little bit of time to kind of squelch the problems if you see whitefly in the field, but you'd have to act very quickly. As far as transmission, again, once the whitefly has the virus in it, efficient transmission to cause disease takes 24 to 48 hours of feeding on the field plants. The virus can transmit it in a shorter period of time, but efficiency is greatly reduced. And then the interesting thing about this virus is it is transmitted for long periods of time by MEM1 or the B biotype of Mycete tabaci. Can be up to 12 days with a gradual decline over that time. The Mediterranean whitefly or the Q biotype, the retention is much less, and the virus is only in the whitefly up to 4 days based on some work by my colleagues in Greece. Larger the population of whitefly, the greater the issue in the field. If you introduce this virus into an area with high whitefly populations, it will infect plants at a very young age. Warm weather increases whitefly movement and dispersal of the virus within a region. And like with CYSDV and other whitefly-transmitted viruses, it can spread into the weeds and also into some other crop plants, leading to establishment of reservoirs that can be used for the virus to be picked up by whiteflies and moved into the crop the next season. If populations are large when plants are young, you can end up with fields like this. This is actually a photo of CYSDV, but the principle is the same, and we have seen the same types of situations for Cucurbit Chlorotic yellows virus as well. CCYV infects at least 13 plant species and 11 taxonomic families. This is very typical of viruses in the genus Cranivirus. Many of the plants that are infected, especially the weeds, remain symptomless. You will not see anything wrong with them. They'll look much like that little mallow plant you see on the right. Could be infected, might not be, hard to tell. Many of these hosts can be reservoirs. Some are better reservoirs than others. The list I have at the bottom, I mentioned alfalfa. Alfalfa is an important reservoir of CCYV. It's a perennial crop. It's grown in many of the areas where melons or watermelons are produced. And as a result, it's an important reservoir for the virus to move. Lettuce, beet, and spinach, although hosts, we do not see them as major epidemiological factors. The virus can infect them, but not particularly well. The weeds are a much greater concern with CCYV. Symptoms of CCYV, as I mentioned, resemble those of a nutritional deficiency, but they also resemble those of other viruses, including, as I mentioned, Cucurbit yellow stunting disorder virus, or CYSDV, Cucurbit aphid-borne yellows virus, CABYV, a different type of virus, but produces exactly the same symptoms. Beet pseudoyellows virus, which is transmitted by the greenhouse whitefly, a different vector, and a number of begomal viruses, including tomato leaf curl New virus and melon chlorotic leaf curl virus, both of which are not present in the United States. They are not in the US, but they look very similar. And it's possible that you could have infection with any of these viruses and not know which was which. Therefore, it's important to have a good diagnostic system, as it has been mentioned before. On the left, you'll see a nice diagram showing how this can work for those of you who may be less familiar with that. I recommend the molecular detection methods because it allows us to detect not only CCYV, but also other viruses that may be present at the same time in the plants in a single test. We can test specifically for each virus using what we call reverse transcription PCR, polymerase chain reaction, or RT-PCR. Quantitative, which allows you to measure the amount of virus, as shown with these little graphs. You can see where the lines come up. We can actually tell how much virus is there, or we can use a plus-minus test. Serological methods are available. They do work. They're better if you know what virus you're targeting. If you don't know which virus you're looking for, there is some cross-reactivity with some antisera, so it is not necessarily ideal for cranivirus detection. The virus can move into weeds, as I mentioned before, and into other crops. So it's important to maintain those weed populations around your fields and in areas near the fields as well, because the virus can move out of those weeds into a new crop quite easily. So manage those with herbicides. If you're going to remove the field from production, treat the plants with insecticide prior to field plowdown. Otherwise, whiteflies will move from the plants that are infected into either weeds or another crop. So it's better to suppress that before you plant. Reflective mulches are another approach that can be used to protect plants early on. As was mentioned earlier, these silver reflective mulches or white reflective mulches are very effective at deterring whitefly in early plants before the rows are fully covered by the vines, and can slow the spread of a virus introduction in the field. Another really nice method that was described earlier is the use of row covers. These Agribon row covers are useful because they can protect plants for quite some time prior to flowering, allow the plants to get some size on them. The bigger the plant, the longer it's going to take for symptoms to develop on that plant when it becomes infected. So you're buying yourself time with these row covers. Once the plants get to the flowering stage, you have to remove the row covers and then treat with insecticide again to keep things under control, because whiteflies generally are very common these days in the summer months in most of the southern US and most other cucurbit production areas in the Mediterranean, parts of the Middle East as well. Transplants are becoming an important part of the cucurbit planting system in North America. As a result, transplants should be infected for whiteflies, eggs, and nymphs, both at the transplant facility and in the field when you receive those plants. Regular testing is encouraged to protect both the grower and the transplant producer. The important thing to mention, as I said previously with the other talk on CYSDV, virus titers remain very, very low for a period of at least 2 weeks. Prior to 2 weeks, it is often impossible to even detect the virus with sensitive molecular methods. Therefore, you need to monitor plants once they get to the field to make sure that you're not having symptoms develop. And if you do, get those tested right away. Ideally, raise transplants in the same region where transplanting will occur so as you don't accidentally introduce virus from one area into another area. Sometimes plants may be infected and you may not even know what's in those plants. There is a source of resistance, or a few sources of resistance, to Cucurbit Chlorotic yellows virus that have been identified in melon. I'm not aware of any that has been identified in other cucurbit crops. The CCYV resistance is currently not yet available commercially, but with time and effort, should be able to complement the CYSDV resistance that's beginning to become available. Just to bring you back to the webpage, the Emerging Viruses in Cucurbits Working Group website has fact sheets on a number of viruses, including Cucurbit Chlorotic yellows virus. We also have the postcards that give a brief overview and show some of the symptoms that you can find on the links in the web page using the QR code shown here at the right, and that will be shown at the end of the webinar. With that, I'd like to say thanks to everybody and let you move on to the next speaker.